What's up, guys? This is Jahangir from MedPorium, and today we're going to be talking about our nephron. Now, our nephron is one of those structural units as part of our kidney that helps filter waste products. Now, this first structure right here, this is known as our Bowmans capsule. So we have Bowmans capsule. This is where our glomerulus and our glomerular capillaries actually meet, and this is where blood will flow through our Bowmans capsule. Now, as you should pretty much already know, blood isn't just red blood cells. There's also plasma, which have proteins, immune cells, and other sorts of stuff. And with our nephron, this is in this region with our Bowman's capsule. This is where we're going to be having these small solutes and water. We could even put like maybe amino acids, or we could put Na or ammonia. So these things are going to be filtered from the blood into the nephron. Now you might be wondering, well, how does blood flow from this glomerulus into the nephron? How do we, start this whole cascade? Well, there are two forces that allow us to do this. These are called the Starling forces, and these are, pressures, but pressure is just force over mass. And these two pressures are hydrostatic, and we are also going to have oncotic. So hydrostatic pressures, they're going to be from the glomerular capillary and from the Bowman's capsule. Same with oncotic pressures, that's going to be coming from our glomerular capillary and from our nephron. Now, number one thing you have to understand is that the reason why blood will flow from our glomerulus, blood will flow into the nephron in this direction right here, going into our nephron, is because our hydrostatic pressure over here is going to be larger than our oncotic pressure. So just to model this, I'll just make that a little bit more smaller. Our hydrostatic pressure is greater than our oncotic pressure, and this allows blood to flow from our glomerulus into the nephron. Now, once we have gotten that out of the way, we can actually see our first structure right here. So I'm going to be highlighting this guy right here, just like that, and something like that. And this is going to be known as our proximal convoluted tubule. Proximal convoluted tubule, or PCT for short. And this is the first region in which our filtrate will actually go to. And through our proximal convoluted tubule, we're going to have these small solutes that we're talking about, amino acids, we have glucose, and let's say we also have some salts over here. So in our proximal convoluted tubule, this is where we are reabsorbing the essential stuff. So one thing you have to understand with reabsorption when we're talking about with the nephrons is reabsorption is when we're going to be taking these good stuff like glucose and taking it from the nephron back into the blood. It'll go through the interstitium, I think that's the correct pronunciation, and this is actually the connective tissue around the nephron, which will go to the vas recta and back to the bloodstream. But just to keep everything simple, basically with reabsorption, I'll put reabsorb. When we're reabsorbing, we're taking stuff from the nephron and putting it back into the bloodstream. And through reabsorption, we're trying to take the essential stuff, because the opposite of that is secretion. So I'll write secretion. So secretion is in which we will have byproducts like waste products like hydrogen, urea, maybe even ammonia, and these are, and also potassium as well. And these are the stuff that we're wanting to take from the bloodstream and put into the nephron, because these are waste products. We're secreting them to the nephron and sort of excreting them out. Because an analogy I like to think about with the nephrons and reabsorption and secretion is, think about it like this. If you're moving to a new house, you're not going to take your expired food or leftover products that you don't even use. You're not going to take that to your new house. But what you are going to take is your bed frame, your mattress, your pillow, lamp, couch, all that stuff, because those are essential. it is not essential for you to take those expired food. Like, you don't need expired food. You're not going to take that to your new house. Those are waste products. So that's pretty much what's happening over here. We're taking our good stuff back from the nephron into the bloodstream, and we're secreting the waste products from the bloodstream into the nephron. So that is our proximal tubule right there, and proximal convolute tubule. And to memorize these waste products, there's a quick mnemonic called dump the hunk: hydrogen, urea, ammonia, and potassium. All right, so, and as you notice over here, we're actually in the cortex of our kidney. Now, if we descend down, we get into our medulla, and this is, let me actually just zoom out over here. If you notice this sort of structure right here, a little bit of a U shape, this is known as our loop of Henle. Loop of Henle. And our loop of Henle is going to be containing two different structures that have different functions. So these include, let me highlight the first guy over here. Let's go with this color right here. I'm going to have to decrease that over here. So this guy right over here, this guy right here, and as you notice, we're pretty much descending. So, cleverly enough, this is known as our descending limb. Descending limb. And the number one thing you want to know about this structure as part of our loop of Henle is that it is permeable and only permeable to water. So meaning that water is the only molecule that can flow through our descending limb, either in or out. Now, the ability to flow in or out depends on our osmolarity of the medulla. So as we go from our cortex to the inner medulla all the way to our outer medulla, the osmolarity is going to increase. So osmolarity will increase. Now, through this increase in osmolarity, we actually have more solutes on the outside compared to inside. If you remember from osmosis, basically water follows the salts. So we have more salts here, so water is going to be flowing into the medulla and back into the bloodstream and whatnot. But basically, water is going to be flowing out, and it's going to be going back to our bloodstream because we obviously need water. We can't just secrete all the water in the world. We still need to keep some in our body. So this is what our descending limb functions as, to reabsorb that essential water that we need in order to function. Now, through this descent, like we were talking about how that increases in osmolarity, the descending limb takes advantage of that. So just to summarize everything that I just said, the descending limb takes advantage of the increase in osmolarity that it's seen in the medulla, and it allows for the higher reabsorption of water. So we basically get to keep more water for ourselves. Now, if we are ascending up, conveniently enough, this structure over here, I might have to move a little bit up, this structure right here is known as our ascending limb. So we're going up. Let me highlight these parts as well and pretty much get up to there, close enough. And in our ascending limb, so I'll write this down, in our ascending limb, this is where we are only permeable to salts. So as you can see, they're both antithesis to each other, both opposites. And a way to memorize this is that descending, think of Dasani. Now, Dasani is a water beverage company, like they sell water. I mean, they did get, there's some conspiracies that they put salt in their water so that you feel even more thirsty and buy more Dasani. But in general, Dasani is water. So when you think about the descending limb, think of Dasani, think of water. That descending limb is only permeable to water, while our ascending limb is only going to be permeable to salts. Now, one thing that you might have noticed is that we have a thin segment over here and a thick segment over here. Our thick segment is called the diluting segment right over here. And the reason why it's thick comes down to the fact that our ascending limb can reabsorb salts actively and passively. passively. So if you remember, active transport is when we need energy. Passive transport is, when we don't need energy. And when we're getting to the active transport site over here, we have a thicker segment because our cells need mitochondria, they need ATP in order to actively transport those salts, because active transport requires energy. Now, one of the key things you have to understand with our ascending limb as well is that as we're going up, osmolarity decreases. So we have decreased osmolarity, and our ascending limb tries to take advantage of that in that we will have a maximum amount of salt reabsorption due to the decrease in osmolarity, and we also have active transport sites that allow us to take in that salt. So basically, ascending limb allows for the reabsorption of salt so we can take it back into the bloodstream. And just as reference, the salts that we're talking about are sodium, potassium, and chlorine, which all have their own important functions for our body. Next, let's move on to our other structure, which we're going to be calling this the distal convoluted tubule. So distal convoluted tubule is that guy right there. So I'll label this distal convoluted tubule, or for short, DCT. Now the main thing you have to understand with this in terms of the MCAT is that there is a hormone known as aldosterone. This is produced by the adrenal cortex, and aldosterone basically allows for the reabsorption of sodium. So basically, sodium is going to be reabsorbed, and by osmosis we know that water follows salts. So when aldosterone acts on the distal convoluted tubule, water is also going to follow our salt. So water is going to be reabsorbed over here, and this basically promotes water retention. So we basically get to keep as much water as we can, depending on how much aldosterone we have, we get to keep that water and put it back into our body. So that's what aldosterone does, and it acts on the distal convoluted tubule as well as the collecting duct. But with the distal convoluted tubule, antidiuretic hormone, which we're about to talk about, does not act. It's only aldosterone is a hormone that acts in the distal convoluted tubule. And obviously, our distal convoluted tubule will have the secretion of waste products like we talked about with hydrogen, urea, ammonia, and potassium. Now let's move on to our final structure right here. And let me get my highlighter for this guy. This is our collecting duct. So this is the region in which we basically get to determine the concentration of our urine. So in the collecting duct, the concentration of urine is determined by the permeability of our collecting ducts. So basically, how much water are we able to be permeable by? How much water can we reabsorb, or how much water that we can not really be worried about? Like, for example, if we're in a hydrated state, if we have enough water, well, the collecting duct will be almost pretty much impermeable to salts and water. So that water is just going to stay in urine, and when you're hydrated, you're not going to see like dark apple juice. You're going to see like a really light yellow color. And if we're in a dehydrated state, well, our body needs to conserve as much water as possible. And there's two hormones that allow us to do this: aldosterone, which we already talked about, and the next one is antidiuretic hormone, ADH. Now, what ADH does is it promotes the insertion of aquaporin channels. So let me get this guy out, and let's just say that this is an aquaporin channel. This is an aquaporin channel. Aquaporin channels just allow the flow of water, so we can get that water back into the bloodstream, and so that we don't suffer those terrible consequences of being dehydrated. But you'll notice that when you're dehydrated, you will see apple juice, because the final concentration of urine is going to be highly concentrated because we don't have a lot of water. We want to keep the water inside of our body in order to basically survive. So all in all, our collecting duct is going to be the one that determines the final concentration of our urine.